All right, so as usual, you can watch this later on the YouTube channel. And let's see, I think we have our quorum. All righty, let's get started here. So welcome everybody to another AP Research Roundtable. Really excited to have Dr. Glennis Logsdon joining us today. And I think unless there's any program notes, Vina, you want to make, I think we should just dive right in to the presentation. Does that sound good?
Sounds great. Hi, everyone. It's good to see all of you. Hope you're enjoying your summer. Hi, Glennis.
Hey.
Excited to have you here today. Is there anybody else joining from Eichler Lab on your end?
No, Evan just went on vacation today, so he's gone for a month.
Okay.
So it's just me.
How nice. He deserves it, as we all do. So I'm glad that he's away. So yeah, go ahead, kick it off.
Okay. Right. So can everyone see my screen?
Yes.
Good. Okay. So hi, everyone. My name is Glennis Logsdon, and I'm a postdoctoral research fellow at the University of Washington in Evan Eichler's lab, but not for long because I'm going to be an assistant professor of genetics at the University of Pennsylvania starting in January 2024. So you can follow me along there, where I'm going to be continuing to do some chromosome AP research. So
Congratulations, by the way. We're all
oh, thank you. Thanks.
Yeah.
So today I'm really excited to tell you about my efforts to build a human reference genome that better represents structural variation on chromosome 8p. And I also want to mention, if anyone has any questions throughout this presentation, just feel free to speak up, and I'll be happy to answer them, because I want to emphasize clarity and make sure everyone understands what I'm presenting. So I bet some of you are wondering, what is even a human reference genome? What is it and why do we need it? So a human reference genome is a set of DNA sequences derived from one or more humans that make up a complete human genome. And the first reference genome was actually built by the Human Genome Project back in 2001. And it's had several iterations over the last 20 years. And it's now what we call GRCH38 or HD38. And some of you might recognize this reference genome because when you get a microarray, this microarray is designed against this reference genome HD38, and it determines all the copy number variations that an individual has, deletions, insertions, and the different types of genes that are present in the genome. But did you know that the human reference genome HD38 is actually incomplete? And in fact, it's missing about 8 to 10% of a normal human's DNA sequences. And these missing regions we call gaps. And these gaps exist in several regions in the human genome, but most notably within the centromeres, which are these regions that exist on every single human chromosome at this primary constriction point, and they're shown here in blue. Additionally, we have gaps in the entire P arm of chromosomes 13, 14, 15, 21, and 22. They're completely missing from this human reference genome. And we also have gaps in these regions called segmental duplications, which are genome-wide. And finally, we have gaps in the telomeres, which you can't really see because they're so small, but they're these little blue regions on the ends of the chromosomes. So together, this is about 200 megabases, or 8% of a human genome that's been completely missing from the established current human reference genome. But in the last 2 years, the Telomere to Telomere Consortium, which I'm a major part of, resolved all of these gaps in the human reference genome. So we generated for the very first time the complete sequence of a human genome, which will now serve as our reference genome. And it's called T to T CHM13. And the reason I'm talking about this now is because the chromosome that I focused on solving was actually chromosome 8. And I generated the complete sequence of this chromosome and resolved 5 major gaps in it, which was published a couple years ago in Nature. So as I mentioned, I resolved 5 long-standing gaps in chromosome 8, and these are important because they are relevant to chromosome 8p disorders. So the first 2 regions are actually the telomeres, as I mentioned, the ends of those chromosomes. The next region, which is super, super important, is called the beta defensin locus. And this locus is located at 8p23, and it's around 7 megabase pairs long. I also resolved the centromere, which is about 2 megabase pairs long, and another region on 8q21, a variable number tandem repeat, or VNTR, that's around 900 kilobase pairs long. But of all of the regions, the one that is most important for chromosome 8p disorders is really this beta defensin locus. And I'm about to tell you why. So the beta defensin locus is one of the most structurally dynamic regions in the human genome. It contains the largest inverted region in the human genome, about 4 megabase pairs long. And so this is a region that flips back and forth in different individuals and across generations. But most importantly, it is the site of recurrent deletions, duplications, and inversions that are commonly associated with chromosome 8p disorders, causing developmental delay, neuropsychiatric disorders, and congenital heart defects. So the beta defensin locus actually causes inverted duplications and deletions on chromosome 8p through a mechanism that was first published in 1996, and it occurs in maternal meiosis 1. So the way it works is that two chromosome 8's sister chromatids line up in maternal meiosis 1. And for some reason, one of them kind of curls over. And then I'm showing here this blue region is where the beta defensin locus is, which has these repeated sequences on either end. And these repeated sequences undergo recombination. And instead of the top one recombining with the top one, and the bottom recombining with the bottom one, you actually get a recombination that occurs between the top of one and the bottom of the other. And this creates a very unique situation where you have a chromosome 8 that is fused to another chromosome 8. And the region over here is kind of popped out as an extra piece of DNA sequence, which is subsequently lost. So when you have this chromosome 8 fusion, it creates a problem in the cell. And that's because this chromosome 8 now has 2 centromeres shown here as circles. And when this chromosome goes into metaphase and begins to be pulled apart, it has a potential for being broken. And this happens a lot in chromosome 8p disorders, where you usually get a break somewhere between D and E segments, where the result is that you have this chromosome 8 with an inverted duplication, C and D, and a deletion of A and B. Okay, so my goal is to really build the first human reference genome that actually has a chromosome 8p structural variant. And keep in mind this current reference genome that we have, HU38 or the T to T1, does not have this represented currently. And this has really limited our understanding of chromosome 8p disorders and how we can effectively target them for gene therapies and et cetera. So my goal is to really build this reference genome that has a complex chromosome 8p inverted duplication and deletion. And the way that I have planned to do this is to generate long-read DNA sequencing data from one of the 8p heroes. And the hero we've chosen is really daughter, a lovely 10-year-old girl who has this complex chromosome 8p inverted duplication and deletion. Once we generate the sequencing data from her, we assemble the data into that reference genome that I was talking about, and then identify and characterize structural variants genome-wide, with the goal of identifying variants that contribute to gene disruption that cause additional phenotypes in the individual. So far, I've completed steps 1 and 2 and part of 3, and I expected about 1 to 2 months more of analysis left for characterizing these structural variants. But ultimately, our goal is to submit a paper on this by the end of the year. So I kind of want to take you through what I've done so far, and also get some feedback. If anyone has any suggestions for or ideas, let us know. But so the first thing we did is really generate long-read sequencing data in the effort to build a reference genome with chromosome 8p structural variation. So the first thing we did is we took this genome and we sequenced the DNA. And we generated 3 different types of DNA sequencing data. The first is called PacBio Hi-Fi data. It stands for Pacific Biosciences High Fidelity data. It's a long-read sequencing data that is highly, highly accurate, and it's incredibly useful for assembling genomes and building these reference genomes. We also generated a similar amount of coverage from Oxford Nanopore Technologies, long-read data once again. We kind of found out that combining these 2 types of long-read sequencing data together really gives us the best result in assembling a genome. And then finally, we also use Illumina data. And this is really to make sure that our assemblies are accurate and representative of what's actually in the cell in the individual. So we then assembled the PacBio Hi-Fi and ONT data into a reference genome. And once we did this, we generated a genome assembly of, and I'm showing you some statistics for these assemblies, which I know don't mean a lot, but I can hopefully give you a little bit of context of what, how good they are. So for we were able to get a set of maternal chromosomes and paternal chromosomes that were each about 3 gigabases long, and this is absolutely expected. You might notice that the maternal set of chromosomes is slightly smaller than the paternal ones, and that's because she's missing part of chromosome 8, which is this little 6 or 7 megabase pair region on the end of chromosome 8. The assembly had something called a contig, which just stands for contiguous sequences. So basically 275 contiguous sequences that we then assemble into chromosomes. And the contig in 50 is about 134, which is extremely, extremely high. So what we have here is an incredibly good genome assembly of with 9 maternal chromosomes that are completely sequenced from end to end, and 7 paternal ones that are completely sequenced from end to end. So what does this look like? So here I'm showing you an ideogram of the assembly from the genome. And I'm showing you all 23 chromosomes. And the chromosomes on top have the maternal chromosome assemblies that alternate in color between blue and yellow. And so this indicates that, so for example, for chromosome 1, we have a sequence that goes almost all the way through chromosome 1 and then shifts to a second contig. So there's a bit of a break here that we need to fix before going back to the 3rd contig that's part of this chromosome. But you can see that there are actually several chromosomes that have complete sequence assemblies. And I'm indicating those with a blue asterisk, like you can see here for chromosome 3. There's a yellow contig that goes completely from end to end. Same with chromosome 4 here. So we have a very, very, very good assembly of genome, among the best in the world, that only needs a few fixes to really get completely resolved. But of course, the chromosome that we care most about today is chromosome 8. So you can see here the paternal chromosome 8 goes from end to end here in yellow, but the maternal one doesn't have this P terminus and instead has this duplication that you can see here in the green and the purple. So I basically, fixed the assembly for chromosome 8 and was able to get complete assemblies of that chromosome for both the paternal and maternal chromosomes. So here I'm showing you the complete sequence of this paternal chromosome, all the way from the telomere on the P arm to the telomere on the Q arm. And I'm showing you here the beta defense and locus. But of course, maternal chromosome 8 has a different structure. And instead of having the same one as paternal, it has this inverted duplication where you basically have this sequence here that's duplicated on the left side, and it also has this deletion. But we were able to completely resolve that. And if you look at these repeat elements that I'm showing you here in this colored track, you can see that this region is basically mirrored on this side. One question we often get is, how do we know that the sequence that we have assembled is actually correct and it's representative of what is in this genome? So to do that, we do several checks, but one of the checks, I should mention this, sorry, mention this, but her maternal chromosome 8 is around 22.6 megabase pairs longer than her paternal chromosome 8. That's about 15.5% of the entire chromosome 8. So to check the assemblies, we align raw PacBio Hi-Fi long-read sequencing data back to the assemblies. And what we hope to see is even sequencing read depth across the length of the chromosome. And that's what we see here for both the paternal chromosome and the maternal chromosome. If there was a misassembly and we made a mistake somewhere, what we would see is an increase in the sequencing depth up towards the 60 number, or a decrease that would indicate we had a misassembly somewhere. But since we don't have that, it really indicates that our assemblies are accurately assembled. Any questions so far? Okay. If we zoom into maternal chromosome 8, we can actually see that it does have telomeric sequences on both the P arm and the Q arm. So telomeric repeats are typically TTA GGG, and that's what we see here, and her Q-arm telomere length is about 8.4 kb. But did you know that on the other side, it's the reverse complement? So instead of TTA GGG, we have AAT CCC, but this exists on every chromosome. So basically what we find here is that it has both of her telomeres, and they're between 6.5 to 8.4 kb long. Just so you all know, a typical human telomere is 2 to 18 kb long. So this is definitely within the normal range of what we'd expect on a typical human chromosome. We can also compare maternal chromosome 8 to her mother's maternal chromosome 8 and define the breakpoints of that duplication and that deletion. So by comparing these two assemblies, we can see that the deletion breakpoint, basically where chromosome 8 has been deleted, goes from chromosome 8 0 to 8.137 megabases in. So she's missing about 8 megabases of her chromosome 8 on the P terminus. We can also define the duplication breakpoints, basically what precise base of DNA is duplicated in. And so this is between 12.5 megabases and 40.5 megabases in reference to HG38. So we conclude that has an 8.1 megabase pair deletion and a 28 megabase pair inverted duplication on chromosome 8p. So once we had, computationally determined where these breakpoints were, we kind of wanted to validate them and make sure that what we had computationally determined is supported by orthogonal methods. So to do this, we collaborated with Mario Ventura and his student Flavia at the University of Italy in Bari. And they performed a procedure called fluorescent in situ hybridization, or FISH. And basically how it works is they design DNA probes to regions of maternal chromosome 8, that either are within the beta defensin locus or on the green or the red parts of the 8p. Hold on a second. So basically, when they performed this FISH on metaphase chromosome spreads from, what they found is that the normal chromosome 8, the paternal chromosome 8, has all three of these DNA FISH probes present, but the maternal one only has the blue one, indicating that this region is actually deleted. So our chromosome 8p deletion coordinates we estimated was between 0 and 8.1 megabases. And the FISH prediction can get as close as it can. It can't ever be perfect, but it's at least 6.5 megabases from the end. We could also validate the inverted duplication breakpoint with FISH. So to do this, we design probes here in the blue, green, and red from the chromosome 8p region that we think is duplicated, and also outside of it, which we do not think is duplicated. And by performing FISH once again, we see that the paternal chromosome 8 has all three probes where we expect them to be on the chromosome 8. But the maternal chromosome 8 has this inverted duplication that we see here, where we have the blue and the green dots or signals being duplicated on the other edge. So this really confirms that the duplication breakpoints are where we think they are, close to 12.5 and 40 megabases into the chromosome. So once we had assembled the chromosome 8 sequence, confirmed the deletion and duplication breakpoints, and validated with FISH, we asked, are there any other variants in the genome? We know there's a very large structural variant on chromosome 8, but what about the rest of the genome? So I worked with a postdoctoral fellow in the lab, Yang Sui, to detect variants genome-wide. And the strategy we used is to really look at single nucleotide variants, structural variants, and indels in the genome with this phased assembly variant caller, PAV, that we developed in the Eichler lab. We then identified variants that are absent in the mother and father, and these are termed de novo variants. They only exist. And we also identified variants that fall within 1,440 candidate genes associated with autism. And so our preliminary results, we identified 7 candidate gene mutations that are associated with autism that fall on, as you can see, chromosomes 2, 3, 7, 10, and 19. But I want to emphasize that these are just candidates, and it's a very preliminary result, and this is not necessarily to be taken as a clinical test. And these are early days, but we're still working on trying to identify these genes. So we're performing additional analyses to hopefully reveal other gene disrupting variants that contribute to autism. So in conclusion, I constructed the first human reference genome with a complete telomere to telomere assembly of chromosome 8. I confirmed the presence of telomeric sequences on the maternal chromosome 8, which are about 6.5 to 8.4 kb long. I refined the breakpoints of the inverted duplication deletion on the maternal chromosome 8 and validated with FISH, and discovered variants across the genome that disrupt genes associated with autism. So what's next? So next, we hope to generate a DNA methylation map of each chromosome 8 to determine how the methylation profile varies between maternal and paternal chromosomes and also across duplicated regions. And we also hope to validate the single nucleotide variants, structural variants, and indels that we've discovered in the genome with native long-read sequencing and other variant callers that we've developed, such as SPEM-ASM and Deep Variant, which was developed by Google. But to kind of put it into context, the broad vision for translating our work to the clinic is we hope to annotate this reference genome with genes and their expression profiles so that this can be widely used as a model for chromosome 8p complex structural variants in a reference genome that can be used for downstream studies. So this would really be best done with RNA-seq or a long-read version of it called isoform-seq or iso-seq from brain organoids. So if there's any other researchers in the Project AP space who are generating this data, it would be really great to map those back to our reference genome and then fully annotate the assembly. The reason we want to do this is that gene annotation and expression would allow us to identify gene targets for therapeutic purposes. So this would be really a great addition to our assembly. And also, we hope to use this reference genome to guide the construction of artificial chromosomes that can model chromosome 8p disorders in cells. So this seems a little bit wild, but my PhD focused on constructing human artificial chromosomes. And I think it would be really great if we could engineer a human chromosome 8 with a similar inverted duplication and deletion and test how this affects gene expression, chromosome segregation, and cell viability. Alternatively, we could engineer a chromosome with the 8p deletion and give it back to the cells with the hope of replacing the missing genes from the individual. So with that, I'd like to thank people in the Eichler lab who contributed to this work, my PI, Evan Eichler, and Yang Sui, who helped with the variant analysis, William Harvey, who helped with the computational assembly, and Kendra and Katie, who helped with the data generation. And of course, the Ventura lab, the two researchers in Mario Ventura's lab who did the FISH, and of course, Project AP and all the AP heroes and funding. And I'll take any questions.
Awesome. Could we go back to the slide with the de novo variants?
Mm-hmm. You're talking about these?
Yep.
Yes.
So these are all by definition heterozygous, right?
Yes.
And I guess, so first question is, what's the expected number of those 1,440 candidate autism gene or candidate genes associated with autism that you would expect, let's say, in a second or third hero if you were to look?
I don't know what the answer to that question is. I think that that's something we could do if we had sequencing data from other AP heroes. I have no idea what the expectation is, but yeah.
A lot? Is this not a lot? Is this completely to be expected? And I get these are associated with autism, but are they associated with autism
AP
at haploinsufficiency? Because otherwise they would be benign if they're heterozygous and not haploinsufficient or not dominant negative, right? So if it's loss of function, then so yeah, I guess that's what I'm trying to get at is yeah, go ahead.
Yeah, I definitely am not an expert in autism genes, so this is why I had Yang help me out with this. But I think the answer would be we need to look at other AP heroes and determine what the frequency is to see if there are any genes that are particularly associated with AP disorders more than the rest of the other autism affected individuals. And then we can actually have the actual empirical test.
I mean, it seems striking. These are all frame shifts. So I guess these wouldn't exist Exact or on any of the larger databases, or are these floating around at some non-trivial?
Right. This is just a subset of the genes that we've identified. We're looking into other ones that are due to not just frame shifts, but structural variants, inversions, that type of thing. But this is a very, very, very early result situation that we don't have a complete list for just yet.
And I guess you also could just tell any gene that whether it was associated with autism or not, whether it has an unusual or private variant. So this list would be much more expansive, just restrict to autism association.
Right.
All right. That was the icebreaker. Someone please follow up with some more questions. I have other questions, but I want to open it up to the scientists in the crowd, or the families. We're actually all scientists here, officially or unofficially. So anyone else want to jump in?
I'll jump in. That was a great talk. Thanks very much, Glennis.
Thanks.
And I'm just curious about, can you take us back to slide 6, please?
Oh yeah, let me see.
I've not seen this 1996 paper before.
Yes.
And so, and like, I'm like, wow, this is incredible. I love how I'm wowed by 1996 research.
Yeah, I know. It's a great paper.
Okay, I'm definitely going to read this one. But tell me, so, because I'm looking at this, right? And I can see, so that's the deletion, right? That goes off on
Yes.
the right-hand side, right?
Yes.
And so then obviously there's a RIP of some sort,
Yes.
somewhere around the capital C, shall we say?
So
Or
so you see this arrow here? It's really
Yeah.
indicating where it happens. So it can actually happen anywhere between the small c and up to E.
Okay.
Yeah.
So then my question is, how on earth does it generate a telomere?
Right. That was actually the surprising finding, that we had a telomere on that chromosome 8p, which doesn't normally exist there. So my guess is that there somehow was a rescue by recombination off of another chromosome's telomere that resulted in it being added on. I actually have absolutely no clue. And because it's TTA-DGG and it's the same on all the chromosomes, I can't actually determine which chromosome it came from, that it kind of duplicated that region over.
Yeah, and I mean, obviously all the children must have telomeres, otherwise they would just,
Right.
they wouldn't be alive. Like they would just be an aneuploidy cell mass and they wouldn't develop in womb, right? So
Right.
they must all have telomeres. And so I'm also very encouraged by the length of
Yeah.
our proband's telomeres as well.
Yeah. It's really positive. Yeah, it's great.
Very good. Okay, thank you. And I still have to sort of mentally process the rest of your talk. And then I'm sure I'll have questions in about a week.
Yeah, no problem. No
I
problem.
got stuck here because
Please email me.
I will do. Thanks, Glennis.
That's a good catch, Faye, in terms of, yeah, if you don't have a telomere, it would not support life. Allison, your hand is up. You're up next.
Oh yeah, it's not really a question, but just to let you guys know that, I mean, we did in organoids both bulk RNA-seq as well as single cell RNA-seq, and this data is available. So feel free to jump in and explore the way you want.
Okay, perfect. That would be great. Yeah. Bina, can you point me to where that data is eventually?
Yeah,
And then
we'll,
I can add it.
we'll send you instructions to access it. Thanks.
Okay,
I don't know if
thank
we have everything, but Whitney will let you know and work with Allison if we don't.
Yeah.
Okay. Thank you, Allison.
Stefan?
Hi, Glennis. Great talk. Thank you. So I had a question about this slide actually too. So, so my understanding was that that subsequently the model was developed that there's a pre-existing inversion on the maternal copy first.
Okay.
And then the crossover mistake. And that this inversion exists in a very sizable fraction of the European population. And there was actually just a paper out that showed the same for, I think, the Japanese population, some like 33% or so.
Right. It's very common. Yeah.
Yeah. And so based on that, so if that's the substrate, right, for the 8p rearrangements to happen, then you would expect that to happen more often if it's such a sizable fraction of the population that carry it. So one of two things must happen, right? Like either that recombination mistake, the subsequent one that leads to the if-dupe del and the bridge fusion bridge cycle that creates the telomere, either that is very, very rare
Yeah.
Or it does happen more often than we realize, but then those embryos don't make it through development. And I'm
Right.
wondering, and I think it's a really hard question to answer, because you would essentially have to like sequence gametes to answer that question. But another way of getting at it would be to look at whether or not, and you have that data potentially, to look at I'm curious about the defensin clusters, the degree of homology.
Yeah. Yeah.
Are 8p heroes more likely to have had a more homologous defensin cluster on one end and the other end, and were kind of exposed to?
So I know a lot about the beta defensin locus. And even though it's kind of simply represented here, she's these little arrows of repeated sequences, it's actually much more complex than that. And in fact, she has 4 blocks of duplicated sequence that are obviously mirror image in her chromosome 8. But this is not normal. It's not normal to have 4. In the human reference genome, HG38, there's 2. In the one that I solved on this chromosome 8, there's 3. And we've done like a survey of different populations. We've seen up to 6 and even 8 duplicated blocks on these edges. And it's, I'm not for sure, but in my mind, I hypothesize that potentially the homology between, maternal chromosomes in these regions might be, predisposing it to be more likely to have this recombination event because of that structure. Perhaps it's more homologous between the two. But we see very different sizes of these repeated arrays on each of these duplication blocks, and perhaps the ones that are more similar are more likely to have that.
I see. And then do you have, and I'm sorry if I missed this, but to identify this extended homology,
yes,
did you do that only on 8p, or did you do it on her mom's 8p? So we know
yes, I did
starting...
solve. Right. So I didn't mention it, but I solved the chromosomes 8 in and her husband as well, completely telomere to telomere. And so I only have one of her maternal chromosomes, and they are very similar. She has 2 duplicated regions, and they are very similar to each other. But I think you're right that it deserves a little bit more attention, and perhaps I can think of some plots that I would generate to determine the sequence identity between those 2 and compare to the husband and see if it might be more likely to contribute.
Yeah, I think that'd be really good for your paper to have that information, right? And
yeah,
it's also good for us to know that, because if there is something rare that, goes beyond just the initial inversion, that might explain why there's so few 8p heroes globally, then it alleviates a concern I had about some of the stem cell models that we're building, right? Because we want them, of course, to replicate development to the closest extent. And so I just want to make sure that, we're not studying something that is, essentially due to a very rare suppressor mutation somewhere else in the genome. So that's really good to know.
Okay.
Great. Thank you.
Thanks. Nicoletta?
Hello. Thank you for your talk. That was great. I just wanted to know, what is your hypothesis in the methylation study?
Oh, okay. Yeah, that's a good question, because I haven't done this analysis yet. I think there will naturally be different methylation profiles between the maternal and paternal chromosomes in some regions. I'm not completely sure what will happen between the inverted duplication regions. I mean, I think it could be possible that they're both turned on, meaning they don't have a lot of methylation over their genes. It could be that the cell has tried to compensate for an overdosage of gene expression by perhaps silencing one set of genes, perhaps this one, or perhaps it varies between the 2. I'm not completely sure yet, and I think it's really going to be an exciting thing to find out.
Like we had some questions in the chat that I think are more of a, that cut somebody off. Go for it. I think there were some questions here having to do with essentially what are the implications of this. So I have a question here about gene replacement for the deleted segments, and I guess maybe expanding on your chromosome avenue.
Oh, yeah. So so there's a lot of research being done now on building these human artificial chromosomes. And right now they're being minimally designed. So what is a human artificial chromosome? An artificial chromosome is an engineered construct, an engineered piece of DNA that has a functioning centromere. And this is the part that I focused on engineering, because if you don't know, if a chromosome doesn't have a centromere, it can't be maintained in the cell and be propagated into subsequent cell divisions. So every chromosome has to have a centromere. So I've kind of built this very minimal circular DNA construct that has a centromere. We found that we can put it into human cells and that it's maintained in those cells across several generations for 6 months. And it segregates just like any other chromosome. And the next step is to really engineer this chromosome to have genes on it. And the idea would be to put genes on this chromosome that are mutated or lacking in the host genome to kind of see this complementary genetic expression of this chromosome. And so that's the next step. But I ultimately plan to really build complete human chromosomes that are synthetic and artificially made, that have custom designed centromeres and genes that are minimal to whatever set that you need. And so the idea for chromosome 8p heroes is they're missing that 8 megabases of sequence on the P arm, right? It's missing right here. So one idea would be to try to re-engineer those into this artificial chromosome and then give it to the cells in order to really express the missing genes that it didn't have before. And the benefit of doing it on a synthetic chromosome, as opposed to, like, for example, engineering it in the human genome itself, is that this is completely autonomous. You can custom design it. What I mean by autonomous is it doesn't affect the host genome. You're not going to accidentally mutate a different gene or disrupt a different gene. It's completely separate, and you can put it into the cell and you can also remove it at will. So if you want to have it express those genes, you can have it, you can turn it off, so it doesn't express those genes if you wanted to. So that's the end goal, is to generate these synthetic constructs that can provide the genes back that the individual is missing.
I guess delivery, the biggest challenge here, as opposed to the genome.
Right, so this is exciting. So in the lab, we have a way of delivering this construct through something called transfection or electroporation, where you basically have a lipid vesicle that contains the DNA construct, and it fuses with the cell and kind of introduces it into the cytoplasm, where it then goes into the nucleus and then is maintained as an extra chromosome. One exciting project is that I'm going to start with Drew Weissman at UPenn. So he was the one who developed the mRNA technology for the COVID vaccines. We're going to work together to basically put these into like liquid nanoparticles and then give them to stem cells, and then hopefully have them regenerate from the stem cells and then produce cells that naturally have them.
Yeah, I was just going to ask if you can just basically do what we did with COVID, but instead of an mRNA spike payload.
Exactly. Yeah. So I met with him during my interview at Penn, which is one of the reasons I'm going to Penn. And he said that we could definitely work together and try to engineer this as a way to deliver it.
So this makes sense for heroes who have deletions only. How would you address the duplications?
Yeah, duplications. So that's a little bit difficult. I wouldn't do it with a human artificial chromosome. I can see using a Cas9 guided approach where you basically delete the inverted duplication and separate it from the rest of the chromosome. And because that little section of DNA doesn't have its own centromere, it will be degraded and lost. And then at the same time, perhaps giving the artificial chromosome with the missing genes back into the cell. So then it's kind of back to, the basal state.
Great. Anyone else want to jump in?
I'm just going to jump in for a second. My eyes are like bulging out of my head. So I want, this is exciting scientific potential, right?
Yes.
So I love the excitement, like from the research community, to kind of think about this, which, but I want to just let the families know, because I have to do my job, is we're very, very far away scientifically, as well as even thinking about this translating to a human safely, right? Or even efficaciously. So just, I think we need to always support this as a foundation. We are very excited about supporting all of this, and always hopeful But just so that the families understand that, you know, this is not near term for our AP heroes. But it's very important for us to continue participating in all of this and having all of our AP heroes honestly represented in some of these studies, because, you know, again, obviously we threw HIP out the window on this call, but is, you know, the case that Glennis has been working on for years now, and it is very hard and challenging, but again, she is unique to her genome. And so that doesn't necessarily mean that all the AP heroes are, you know, identical to this conversation. So it's really important for us to better understand that.
Yeah, and I saw there's a question in the chat about how someone else can get their AP child involved. If you want to get involved, this was just a pilot project on one family, but we're hoping to expand it to have multiple different reference genomes. So please email Bina or me and let me know. And then perhaps we can put you on like a priority list to be like the next reference genome that we build.
Glennis, for that, I know you guys also wanted, I did send the community a note that if they had a proximal deletion only.
Yes.
And so we have some interest on that, but right now your kind of request, is that for proximal deletion only or a specific type of genetic rearrangement?
Deletions are a good start because it's completely different from this genome, meaning there's no inverted duplication. But I think that other variants of the inverted duplication deletion would still be of use, especially when it comes to DNA methylation and gene expression, and seeing how those are kind of balanced in different contexts.
Okay, that's good to know. So guys, I mean, I really encourage the families, if you guys like have no idea what any of this was about or how this applies to, you know, the overall mission of AP or specifically clinical questions, I mean, just ask them and we can say like, we don't know, or Glennis and the team will try to respond to that.
Any other questions or comments?
I'm just gonna echo what Bina said. I like completely agree about, you know, it's brilliant work, but who knows when we could possibly see it going into heroes. I think that, you know, I sort of allude to it in the family-friendly research talks that, you know, I think an absolute best case scenario would be 10 years, and that's if all the stars aligned, you know, et cetera, et cetera. So that's a, you know, a sort of ballpark if everything went according to every wonderful plan ever. And I've never known science to work like that. But miracles do happen in science, and that's why you can never kind of like give up hope either. One comment to Glennis as well, though, I do know there are, and Bina, maybe you can fill us in as well. I think there are a few families who have that ring AP or a ring.
Oh, cool. I love ring chromosomes.
Yeah, and so I wonder if you studied some of them, you know.
No, I haven't. They would be wonderful to look at, as well as the other ring chromosomes that exist in the genome, ring 14 and 20.
Exactly.
Yeah, it would be great because actually there's beyond understanding like the etiology of those chromosomes, they actually help me understand how I'm building my artificial chromosome, which is also technically a ring. So it's helpful to understand how ring chromosomes, you know, duplicate, segregate, are maintained in the cell, and what their genic content is.
And presumably don't generate telomere. That's how they solve their enzyme problem, right?
Exactly.
They turn into a ring.
Right.
So yeah, no, totally. That would be a nice sort of like thing to study alongside if you can get any of those. I would offer my proband, but whoever it is Madison, but I don't believe she has rings. So ring was a mate. Thank you.
Hey, Rui.
Hi, that was a wonderful talk. Really great work. So I guess I have a few questions, but the first one is when you look at the centromeres, do you have you? So one of the things, maybe I can start. One of the things we're really interested in is trying to figure out why certain individuals are mosaic for you know, Down syndrome or AP or what have you. So we've been trying to look into the centromere sequences because we believe that missegregation continuously happens because there's some, you know, sort of rearrangement or something in the centromere. So have you Is that something you're interested in?
Yes, absolutely. So actually, my true expertise is in centromeres, and was one of the people who completely resolved their sequences in the human genome. And we've actually been looking, I'm on the same like wavelength as you. We think that variation within the centromere, either at the genetic level or the epigenetic level, contributes to missegregation. And we've been looking at its role in Down syndrome or trisomy 21, where we had actually hypothesized that variant centromeres are more likely, on chromosome 21, are more likely to make them missegregate. And so I can tell you a little bit about what we've done so far. We sequenced an individual with chromosome 21 trisomy, looked at its centromeres relative to its parents, and we found that one of them is abnormally small, about 10-fold smaller than the other centromeres. And on top of that, the small centromere has a really different and unexpected DNA methylation profile, which is kind of marking the location of this multi-protein complex called the kinetochore, which is really important for chromosome segregation. So just from our early studies of this one individual with Down syndrome, we actually do see very obvious variation that's probably likely contributing to the chromosome missegregation frequency. But we're now in the process of looking at 40 other individuals with trisomy 21 to see if this pattern holds true, that they have abnormally small centromeric arrays with perhaps abnormal DNA methylation statuses that can contribute. So we haven't yet validated it, but we're hoping to find a pattern in the others.
Very cool. I think that would tell you why they got trisomy 21 in the first place.
Right. Yeah.
So we have a large cohort of mosaic samples. We're trying to figure out how these individuals continue to lose it throughout their lifetime.
So
Right.
mosaic samples are a little bit harder for us to work with from the genome sequencing side because there's mosaicism, like you mentioned, like it makes it difficult from the computational part to separate out the mosaic cells from the non-mosaic cells. I mean, I guess what I mean is those who have a trisomy 21 versus a diploid 21. But so it's a little bit difficult. And we had originally thought about trying to go after mosaic chromosome 21 trisomies, but we kind of thought, let's just go for, a complete trisomy 21 first, and then perhaps as computational algorithms progress in terms of sequence and assembly, we can get to the mosaics. But they are really difficult for us right now.
And this is because you're using patient samples instead of cell lines that have been established from those individuals.
So right now we're using a cell line that's available publicly, but we actually haven't gone to blood samples from patients just yet. I think that would be a next step.
Yeah, but we have the cell lines from mosaic individuals with both euploid and trisomy separated, right?
Yes. Okay.
Yeah. So
you have them separated. Okay.
Yeah,
okay. That makes them easier. That's definitely more, easier. Yeah, I see what you're saying. Okay. So basically then you could sequence and assemble one of the sets or both sets separately.
Correct.
Yeah.
And we have one for AP.
Very cool.
Which is also going to be separated, or it is separated. We're just trying to figure out what it is. Cool.
So I think it would be great to if you email me after this, and then we can talk a little bit more about it, because it actually seems like a very now more tractable problem that we could potentially solve from the mosaics if they're separated out. It solves a lot of our problems.
Cool. Thank you.
Mm-hmm.
All right, I think we got everyone or many folks engaging today on the call. So good testament to the quality of the research presented, Glennis, and all the sort of creative sparks it's generating. Any other final questions or comments?
I mean, I'll just add on the research side, this project that I think some of the scientists on the call are aware of is like this kind of partnership we've had with Illumina to at least get sequencing data, and the raw data sets will be shared. They actually have all their short reads complete and some of their synthetic long reads. They will do the DNA methylation. It's going to be on all the AP heroes that have given their blood samples. You know, and so there is like some process and consenting and registering and coordinating all of that if you have not been able to do that for families on the call. But that data, it might be interesting, Glennis, for you to have.
Yeah, I think it's a good start. And then perhaps from there, we could pick certain individuals that we would want to do long read sequencing with, like PacBio and ONT, and then, you know, get some really complete assemblies. But I think it's a good way to start for sure.
Great. So I know that there's like been some guidance here from some of the other collaborating scientists on AP. So, you know, we really appreciate your input, and Glennis, like, we'll follow up with you on some of the, you know, suggestions made here, because we'd love to, you know, incorporate that prior to you completing the full analysis.
Cool.
and publishing. I know that we're just eager to have that also published, because it'll help us move some other things forward too. But we're excited for your new move too. And I know that you'll be collaborating with Eichler and some of their efforts, you know, ongoing. So yeah, it's just building, I think the biorepository will be important for us. And so Stefan, thanks again for like your really helpful questions. Faye, go ahead.
Oh yeah, I was just gonna, sorry, I didn't mean to interrupt. I was waiting for you to finish. I just wanted to say to the families on the call, like, you know, if you guys have questions and really just want to understand, please reach out to Katie and just let her know, and she can set up a meeting, you know, and we can get to the point where we can get your questions so that they're, you know, if I can't help you understand, we can together, you know, work with our incredible researchers here and Ethan, and we can make sure that, you know, you understand, because it's really important that we all do this together.
Yeah. And then I think we answered some of your questions, but your last one in the chat about discoveries specifically for getting applied. I mean, you know, she's kind of like been the volunteer pioneer, or, you know, beta test, whatever you want to call it, because we had her skin cells and we had some things on her. So the researchers were able to start that almost like pre-COVID. So then things halted. So, you know, application for all of this is that we learn a lot for how this relates to the broader AP community, and better understanding the biology and better understanding what's really here. It's not going to necessarily be specifically applied for her, frankly. But yeah, it's just, you know, as much as we can have a representation of this very dynamic population, all of our kids, like we say, there's heterogeneity, which means all of the symptoms vary, right? There's mild to severe things across every organ system. And so we really do need to understand how each hero is a part of that bigger puzzle, so we can solve the problem collectively. I don't know if, Lynn, that helps you, or if you have more questions.
No, that's helpful. Also, I'm not.
Yeah, we can hear you.
We can hear.
Okay.
Yeah.
When whomever addressed it said this, you're very much at the beginning of the research, and, you know, short-term, long-term, at least 10 years, and we see what happens. But ultimately, is that a result that you're looking to, that you will be able, like putting stem cells in people, be able to put in the revived or redesigned, I'm not absolutely sure all the correct words, element into a child, so that some parts of the AP can be addressed?
It is a goal. We're going to do our due diligence of starting small, start with cells, start with a petri dish and try to get to a cell line, and then do some testing in macaques. That's the plan, and see if we can get this to work. But ultimately, that would be the goal. Yeah.
All right. So keep up the good work.
Thanks, Lynn.
And I'll add to that. I mean, you know, another thing we're also really putting our energy and efforts behind, driven by Ethan Perlstein as our research director, is the repurposing of drugs. So that's a little easier, right, to kind of think about that. And Nicoletta Moisei's lab too is just kind of looking at, are there existing drugs that could be used that we know are safe and efficacious, maybe for other diseases, also on AP, and would that just help improve some of the symptoms? So it's not, you know, curative by any means, but and then we could use some of this information, like what Glennis just presented, to maybe help validate, you know, that this genetic rearrangement is accurate and precise, and then use some of our other researchers like Dr. Maharena's work on the 2D neuronal cell models, or Dr. Motri's work on the organoids. Like, could we see an improvement in the dish again on those disease models, just for some drugs that are already on the market that we have access to? So, there's other approaches out there that we are, thinking of and are putting resources behind on those strategies.
Great.
All right, that was a stupendous AP Research Roundtable. Thank you, Glennis,
Thanks.
for keeping us in rapt attention. Thanks everyone for engagement. We had real stellar engagement today. Let's keep that up and keep this momentum going through the rest of the year.
Awesome. All right.
Thank
Thank
you,
you
everyone.
all.
Great. Good.
Thank you all. Bye.
Bye-bye.
Bye.
Bye.